All right, so let's take a look at scenario number 2. It says you are asked to discover in which temperature amylase functions best. Amylase is an enzyme that breaks down carbohydrates into simple sugars. So again, I'm going to highlight that because that seems like important information. It's describing the function of amylase. You set up 3 different beakers. The first has a solution with 15 milliliters amylase, 10 milliliters carbs, and is in the freezer at 32 degrees Fahrenheit. The second has a solution with 15 milliliters amylase, 10 milliliters carbs. It is set out on the counter at 70 degrees Fahrenheit. The third solution with 15 milliliters of amylase and 10 milliliters of carbohydrates is on a hot plate at 100 degrees Fahrenheit. So again, let's draw a picture so we make sure we understand what we're looking at here. Okay, so we have 3 beakers. And if I look back at the description, it looks like they actually all have 15 milliliters of amylase, 10 milliliters of carbohydrates. We can see all 3 of them have that same amount. So let's go ahead and put those amounts in here. What was it, 15 and 10? Yes. ml and then 10 carb. Okay, and then so the first one was in the freezer at 32 degrees Fahrenheit. The second one was at room temperature, 70 degrees Fahrenheit. And the third was on a hot plate at 100 degrees Fahrenheit. Okay, so let's see if we can use this information to identify our groups and variables. So what is the independent variable? What is different between these 3 things? So the amount of amylase is the same, the amount of carbohydrates is the same, but the temperature is different. So we're going to say temperature. Okay, what are we measuring? So it says amylase is an enzyme that breaks down carbohydrates into simple sugars. And we're putting carbohydrates into each one, so I'm going to guess that we're going to measure the amount of simple sugars at the end. Because if there are simple sugars, that means amylase did its job. Controlled variables. What did we not change between our groups? We pointed this out earlier. We have the same amount of amylase and the same amount of carbohydrates or substrate in each container. So amount of amylase and carbohydrates. So the enzyme and the substrate. What's our control group? So normally we would say our control group is 0, but remember with temperature we don't really have 0. 32 degrees Fahrenheit is freezing, that's pretty cold. 100 degrees Fahrenheit is pretty hot. 70 degrees is more normal, so that would be our control group. And then the other 2, of course, would be our experimental groups. All right, it says write a hypothesis. What do you already know about how temperature affects enzymes? Which temperature do you think the amylase will work best in? Why do you think that? Write at least 4 sentences. Okay, I know that heat denatures enzymes. I know that cold temperatures make the molecules move slowly. So I'm going to think about amylase. We read about that before. Salivary amylase. Remember? That's something located in the human mouth, in saliva. So I'm thinking that amylase probably would function best near human body temperature, which we know is 98.6. So I think amylase will function best at 100 degrees Fahrenheit because that's close to human body temperature. At 32 degrees Fahrenheit and 70 degrees Fahrenheit, I think the molecules will move too slow. So something to point out here, guys, you may not have the exact same hypothesis as me, but as long as you used background information, which is right here, a prediction, right here, and then reasoning, because that's human body temperature. As long as you have background evidence, a prediction, and reasoning, you're good to go.